Hello everyone, my name is Anthony Newman and I'm a publisher at Elsevier in the Netherlands. I'd like to welcome you to this webinar being hosted by the journal Clinical Mass Spectrometry and entitled Therapeutic Drug Monitoring of Antibiotics. Thanks to our sponsors SCIEX for making this event possible. Clinical Mass Spectrometry is the journal of the organization MSACL, Mass Spectrometry in the Clinical Laboratory. MSACL, as well as creating this journal, also holds annual conferences in spring in the US and in autumn in Europe. The combination of these high-quality conferences and this journal provides a great platform for researchers to learn about the latest innovations in the field of clinical mass spectrometry. As part of its remit to provide educational resources for the community, Clinical Mass Spectrometry is also organizing a series of useful webinars around mass spectrometry in the clinical laboratory. And this is one of those. In this webinar, the speaker will discuss and present therapeutic drug monitoring, TDM, of antibiotics. Antibiotic resistance is becoming an ever-growing threat to healthcare delivery. Both antibiotic misuse and insufficient dosing are the two main reasons for resistance emergence. Given the lack of novel antibiotics under development, optimized drug exposure is essential to suppress the spread of antibiotic resistance. In the past, therapeutic drug monitoring, TDM, has been employed only for antimicrobials with narrow therapeutic ranges, such as aminoglycosides. However, in recent years, the knowledge about the altered pharmacokinetics in critically ill patients has led to a paradigm shift. Nowadays, TDM of antibiotics is also being advocated as a tool to guide dosage, helping provide adequate drug exposure and therapeutic success in patients with severe infection, and at the same time minimizing antibiotic resistance. So accordingly, antibiotic TMD or TDM is becoming more widespread and increasingly becoming recognized as a core element of hospital antibiotic stewardship programs. So this webinar will review the basic requirements, the indications and practical aspects for rational antibiotic TDM in the intensive care setting, with a special focus on the beta-lactams, the most widely used antibiotic class. So it's my pleasure to introduce and welcome today's speaker, Michael Pahl. Dr. Pahl is currently working in the Institute of Laboratory Medicine, Laboratory of Mass Spectrometry at LMU in Munich. His special area of interest is therapeutic drug monitoring of antibiotics in intensive care medicine, so he really is the best person to speak to us on this topic. LMU is also rather special. The Ludwig-Maximilians-Universität Munich, or the LMU, is one of the leading research universities in Europe with more than a 500-year-long tradition in research and teaching. Before we begin, let me remind the audience we'll have a question and answer session following the presentations. And please submit your questions any time during the presentation using the Ask a Question button on the right-hand corner of your screen. I would encourage you to input questions as and when you think of them, and these will be addressed in the question and answer session at the end. The more questions asked, the better the session will be, of course. And also, as a reminder, in a few days' time, this webinar will be available as an entire recording, including all the questions and answers online. So without further hesitation, I'd now like to hand over to Michael Pahl to begin his presentation. Dr. Pahl.
Thank you, Anthony, for this very kind introduction. So, again, to say it, my name is Michael Pahl. I'm a doctor at the Institute of Laboratory Medicine at the University Hospital of the LMU Munich. And my main area of research is the antibiotic TDM, where I'm in very narrow contact with also our physicians of the intensive care units. And we try to make the best out of it. So in this world of an ever-growing emergence of resistant bacteria. So as you can imagine, the field of TDM of antibiotics is a huge one. So I'm just going to address from my point of view, most relevant aspects. First of all, why we have the problem of an increasing antimicrobial resistance, the causes, and why it poses a threat to global health. The second point of this talk will be the so-called pharmacokinetic and pharmacodynamic aspect of antibiotic TDM. because it's, well, we need to do some pharmacokinetics just to know why the physicians are aiming at certain dosage or concentration levels. The third point will be the so-called paradigm shift of antibiotic therapeutic drug monitoring. As Anthony already said in the introduction, in the past, antibiotic therapeutic drug monitoring has been mainly employed for the drugs we call most toxic, which are the glycopeptides and aminoglycosides. But nowadays we know that it is essential also for the so-called non-toxic antibiotics, especially the beta-lactams, which are the most widely administered antibiotics at all. Then we will discuss another point, which is the antibiotic stewardship. So what are the fall pits? What needs to be done in a local hospital? Another point will be the quantification techniques. So how can antibiotics be quantified nowadays? What is the state-of-the-art technique to do antibiotic TDM? And finally, the challenges for the future. So what can we do? What can the IVD industry do to make things easier for the physician and also for the laboratory analysis? So let me get to the first point, which is the antimicrobial resistance. So why has it become a threat to global health? So the main reason why we have an emerging antimicrobial resistance is simply said, it's just the overuse of antibiotics. So antibiotics have been on the market or had been used for the last few decades, and the main problem is simply unregulated use. Many antibiotics are easily accessible. You can also get them from the online pharmacy. And another big problem, of course, is the extensive use also in livestock farming. There are also certain studies where they say that even physicians in up to 30 to 50% of the cases that they have an inappropriate prescribing procedure, which means you either have too high concentrations of your antibiotic in your bloodstream or the concentrations will be simply too low. And whenever I have a pathogen and I can't eradicate it, so this will also contribute to the emergence of resistance. And another problem is, of course, when you're treating a disease or an infection and you don't know the pathogen you're fighting or you don't know the resistance pattern. This will, of course, also fuel the infection, the antibiotic resistance problem. So regarding also the prescription of antibiotics, on this slide you see a prescription scheme from the 2010 in the United States. And usually you would expect that if you have the same distribution of diseases in a country, that you would almost have the same prescription pattern. But what you can see here, for instance, is that in certain states you have a higher prescription compared to other ones. And this, for instance, could be attributed to this could be, for instance, indicate that there could be an improvement in the antibiotic prescription with fewer unnecessary prescriptions. And this, of course, then would also be a better situation regarding the emergence of antimicrobial resistance. We also see a same pattern almost in Europe, where we see that from the countries from north to south and west to east, that we have higher prescription rates in the southeastern countries in Europe as well, which would also fuel antimicrobial resistance. Let me show on the next slide also what is the current resistance situation we face in Europe. You see, for instance, on the left slide, the resistance of gram-negative pathogen Klebsiella pneumonia for third-generation cephalosporins from the data from 2005 and 2011. And the darker the color gets, the redder it gets. You simply see that there's an increase in resistance. And as I have said before, you see some kind of east-west and north-south divide. So we clearly see also from the data in literature that resistance is increasing dramatically. What we see from different isolates across Europe, we can also observe in intensive care units. And here is also especially relevant because these patients, they are very ill, they are critically ill, they are very vulnerable. And when you even face resistance problems at the intensive care units, where they also have access to the to various antibiotics, to almost every kind of substance which is available on the market, this is really a big issue. And let me say this as well, that in a recent survey from 2011, almost every second infection specialist said that he encountered in the last year one pathogen which is pan-resistant, which you would call, or the common people call, superbugs, which means that this kind of pathogen is not susceptible to any kind of antibiotic, which means that the prognosis in these patients is severe, and that an infection which such a kind of pathogen will almost, in most cases, cause death. So what we see on the resistance side, we also see in daily life, which means on this slide you see the so-called daily units, which is disability adjusted life years in Europe, which is some kind of surrogate marker which expresses the years of life a person will lose due to disability, an illness, or even death. And what you also see that, well, as the resistance increases, almost the dailies increases as well. And in this kind very nice publication, which was published in The Lancet this year, they even state that each year in the European Union, more than 30,000 people are dying due to the infection with a resistant bug, which could be those superbugs. Yeah. And they even estimate that in 2050, more than 10 million people worldwide will die due to resistance bugs each year. So antibiotic resistance is really an emerging problem. So, and we need to face it now. So there's another bad starting point which we got right now, which you see on this slide. The antibiotic pipeline looks bleak, which means most antibiotics we are using right now in clinics and which have been approved, they are rather old antibiotics, and we don't have a lot in the pipeline, which means we have to preserve the resources we have got right now, and we can't expect major breakthroughs by approval of newer antibiotics. And you see it also on the right side, which means that approval rates for new antibiotics, for new antimicrobials, they have been decreasing over the last years. So why is that? I mean, we see a problem. We know that resistance is an ever-emerging problem. So why does the IVD industry and the government, well, why is there no bigger effort to generate or create new antibiotics? Well, a big portion or a big problem of all of that is just the finances. On the left side, you see the sales rates of recently launched antibiotics. For instance, Teflaro is cephalosporin, and exactly it's the ceftaroline prodrug compared to other pharmaceutical drugs on the right in the image B. And yeah, the problem is simply that pharmaceutical companies, they make the biggest income with drugs for chronic disease, like Januvia, which is also sitagliptin, which is an oral anti-hyperglycemic drug, or for instance Lyrica, pregabalin, which is used for seizures and neuropathic pain. But antibiotics, well, they simply have a rather low income, so which means it's not so interesting for companies to simply produce antibiotics to get an approval. And what you also have to think of, that when you're treating a patient with an infection, you are aiming to cure him, which means you are only prescribing or administering this kind of substance for a limited period of time, just compared to a chronic disease, chronic illness. Here you have a patient, you have a customer which needs the drug for the rest of his life. So there's really a low return of investment for the antibiotic substances. And another point is also when the physicians are administering antibiotics, whenever there's a new antibiotic which is very effective against a pathogen, they often keep it as some kind of ace in their sleeve, because they say it's so precious, I don't want to waste it on a common infection, so I will just administer it whenever there's no other choice. So even that is also giving a low return of investment for pharmaceutical companies. So let me now show you the key aspects of the so-called pharmacokinetic, pharmacodynamic target attainment. So the basic principles also of pharmacology. So when you think of a drug and you're giving it to a patient, there are two effects which will result in some kind of which will give the pharmaceutical effect. First of all, it's the pharmacokinetics. So what is your body doing with the antibiotic? On the right side of the picture, you see there are several steps. First of all, your drug needs to be absorbed. It needs to distribute within the body and also reach the site of infection. Then it's going to be metabolized, and then it's going to be removed from the body by clearance, which is the excretion step. So this kind of pharmacokinetics, it influences how much effective drug will be there in your body. The second point, which is also relevant for the effect of your drug, is of course the so-called pharmacodynamic effect. So how will your drug, for instance, bind to a receptor? What is its efficiency to eradicate pathogens? So those 2 points, well, both together, in the end, they give you your effect of your substance. When you think it over from the pharmacokinetic aspect, what you see on this slide is when you give an antibiotic or any kind of drug to a patient, it takes some time until you reach the so-called steady state. So what you see on the slide with the pill symbol, you give a drug to a patient, so it will be absorbed, it will distribute in the body, and then you reach a so-called peak level after a certain period of time, which is usually 30 minutes after administration, but depending on the way you give it, if you give it orally, it could also be reached 1 to 2 hours after administration. You will reach a certain peak level, and when the drug is being excreted from the body, you will reach again the so-called trough level. And what we usually have in the clinics, we say that whenever you want to measure those kind of levels, the peak level and the trough level, you need at least 5 half-times until you reach the so-called steady state, because this is now a state where you have rather stable trough levels and peak levels, which is the best time to perform therapeutic drug monitoring of any kind of substance. What is also essential is to know what is the so-called area under the curve. So the area under the curve is simply the integral of the area between those kind of 2 trough levels and the peak levels, which means whenever you want to do some kind of TDM and whenever you need to record a kind of area under the curve, it is clear that you need multiple drugs, multiple sampling of blood in order to get your area under the curve. So the area under the curve, it has the main advantage is that it gives you the average concentration of your drug over time, and then also it simply highlights what is the kind of clearance you have for your drug. So you get the most information when you want to record the area under the curve, but it's also the biggest effort because you need multiple drug sampling of blood. So for antibiotics, the key parameter for treatment is the so-called minimum inhibitory concentration, MIC. In German you would call it minimale inhibitorische Hemmkonzentration or the Hemmkonzentration, which is the MHC, which is the key parameter, and it's simply a drug concentration for a specific pathogen, where it states that when you expose the pathogen to this kind of concentration, as shown in this image by serial dilution of your antibiotic, then you won't see any kind of visual growth. Which means if you have a lower MIC, your drug your pathogen is very susceptible for a specific drug. If you have higher MICs, your pathogen becomes more resistant, and then you need higher amounts of your drug. Then it could be even advisable to simply change the antibiotic and to choose another one where he's susceptible. So what you have to consider that this kind of MIC, it will be dependent of the drug you're using, but also on the pathogen at the site of infection. So I placed it as some kind of local pathogen. And what you also have to consider that those kind of MICs, that they might vary between the laboratory, but also between the operator. So you always have some kind of intra and interlaboratory variation. So what are you going to do when you have no MIC available? Let's assume you're in a rather small hospital. You don't have any kind of microbiologist which can give you some help. What are you doing then? Well, you could use, for instance, the UCAST clinical breakpoints. So UCAST is the so-called European susceptibility testing with the clinical breakpoints, and you can use those breakpoints for the PKPD, which is non-species related. So which means when you are trying to fight a pathogen, you are addressing those kind of breakpoints. They are usually higher than your specific pathogen you will face, but they are the targets, the so-called MIC targets you should use whenever you don't have a MIC available for your specific pathogen. So we have been discussing about the pharmacokinetics. Another thing which is very important for the impact of a drug is the pharmacodynamics. So what does the drug does to the body, or let's say the pathogen? So when we see antibiotics, we have to distinguish between antibiotics which have a mainly time-dependent effect and compared to other ones which have mainly a concentration-dependent effect. So if you have a time-dependent antibiotic, you want to keep the concentration for the duration of your treatment over a certain threshold. And this threshold would actually be the trough concentration. So for time-dependent antibiotics, you would do the antibiotic TDM using blood sampling at trough concentration levels, which is usually the time point before you administer the next dosage of this specific antibiotic. If you have instead concentration-dependent antibiotics, you want to achieve concentrations as high as possible, which is depicted here as Cmax, and you want to measure your concentration at the peak, which means you're going to measure it usually half an hour or 1 hour after you administered an antibiotic. And of course, there's also some combination. Certain antibiotics have a time-dependent effect and also concentration-dependent effect. And for these antibiotics, it's advisable to use the area under the curve for the therapeutic drug monitoring. But as I said before, this takes a little bit more effort because you simply need more blood samples to generate an area under the curve. So, just to summarize it up, the pharmacodynamic effect of an antibiotic is the ratio of your free drug, which means the drug which can also exit your blood vessel stream to reach the site of infection, to the MIC, which means when you have higher concentrations of your drug, and if you have higher concentrations compared to a MIC, this is a very good thing. You will reach concentrations which are necessary or which are sufficient to prevent growth of your bacterium. So the main ratio or the main pharmacokinetic and pharmacodynamic target attainment is the ratio of your free drug compared to the MIC, whether it's the trough level or the maximum level or also the AUC, the area under the curve. So this next slide shows you the so-called Mississippi mud. You can see on the left side that you have some kind of brownish substance, which on the right side was turned rather whitish. It's vancomycin. So initially, the therapeutic drug monitoring was performed mainly for drugs which were considered toxic. If you watch this picture of vancomycin preparations, you can imagine when you're infusing a patient this kind of brownish substance where we have various compounds, that you also have some rather toxic effects. And this was also the case for vancomycin. So the early formulations, which was called at that time Mississippi mud, they caused severe kidney failure and also had some toxicity to the inner ears. So it was not astonishing that therapeutic drug monitoring at that time was introduced for vancomycin simply to prevent this kind of toxic side effects. If you compare it to one of its predecessors, the teicoplanin, there, for instance, this drug was created semi-synthetically, and there wasn't actually necessary to perform at the time therapeutic drug monitoring, or it was not considered necessary, but nowadays we have it again. We are performing therapeutic drug monitoring for all kinds of glycopeptide antibiotics, which means we simply have a kind of history, what was worth of performing therapeutic drug monitoring and what was not worth of doing it. So there has been some kind of paradigm shift in history, especially for the beta-lactam antibiotics. What you see now on this slide is what I have shown you previously. When you're trying to administer an antibiotic to a person, you will reach a certain trough level, peak level, and so on and so on. Let's assume you have a change in pharmacokinetics, you have a smaller distribution volume, the patient has a smaller clearance of the antibiotic. You will simply follow the red line and you will reach levels which are considered toxic. So at that time, mainly TDM was performed for so-called toxic antibiotics, let's say glycopeptides and aminoglycosides, especially when they were given in combination. But what we face nowadays is also a problem about the green line, which means, let's assume you have a higher clearance of an antibiotic and now you reach subtherapeutic levels, which means you don't have a therapeutic effect of your antibiotic, which will also fuel the emergence of resistance. And this is especially true for the beta-lactams, which are the most widespread used class for therapy of ill patients. So nowadays, we use also the TDM for checking whether we have toxic effects, still for the glycopeptides and aminoglycosides, but we are also looking for patients which have subtherapeutic levels. So when I learned medicine, well, the average patient, it had 70 kilograms, it was young, often male, and think the same of when they did the pharmacokinetic studies. So they did those kind of studies for the various drugs with mainly young people which were not ill, which had a rather good physical condition. And of course, in the days of today, we have simply different patient collectives. So we have patients, older patients, we have patients with several illnesses. So those kind of pharmacokinetics, they are not valid in those kind of patients populations, which means the beta-lactams, which were at once at a time considered safe with a regular administration regimen, now we see that many patients which receive the regular dosage, that they simply don't reach the concentration level they should have in their bloodstream. And why is that? On this slide, you can simply see that we have, for instance, altered renal clearance, which means you have a bigger elimination of your antibiotic. We have, for instance, also an increase in the volume of distribution. For example, with capillary leakage, also the antibiotic can exit the bloodstream, so there's less antibiotic concentration at the site of infection. And we have also external factors, especially in critically ill patients. When you perform extracorporeal theories, whenever you attach a filter, when you want to get rid of a certain substance and you are filtrating your blood, you will also lose your antibiotics. So those are especially the patient populations where you really should perform antibiotic TDM. On this slide, you see for the beta-lactam antibiotics the targets which should be reached. So whenever we talk about targets which should be reached, we got this data actually from in vitro studies and also in vitro studies and also animal experiments. So many people consider a concentration of free antibiotic substance larger than the MIC for the entire duration of the therapy, which is the third box from or the gray shade from the coming from top to bottom. They consider it adequate for therapy. And actually, the 50% free antibiotic substance over the MIC this is really the absolute minimum which you should have in your bloodstream of a patient whenever you are performing TDM. You see the green arrow. What we are actually aiming for at our institute, our hospital, are concentrations of trough levels for the beta-lactam antibiotics which exceed the MIC by 4 to 5-fold. So I will also explain you later why we do so. But now let me show you a study which was very interesting. It's the so-called DALI study, which was performed by Roberts in 2014. So DALI stands for Defining Antibiotic Levels in Intensive Care Unit Patients, which was some kind of prospective study where they got data from several intensive care units, and they simply tried to check, well, do the patients, when they receive regular dosage, do they reach the levels they should have, and what is the prognosis, what is the outcome? And what you see on the top in table number 3 is that many patients even did not achieve the minimum required concentration level, which is the 50% of the free antibiotic substance, which should be for 50% of the therapy be higher than the MSC of a specific pathogen. So you can clearly see that with dosages which have been established in the past for the people which are not critically ill, which are not so susceptible, that these dosages are simply not sufficient in many patients. On the bottom you see also another slide where they simply compared, well, what happens if a patient has a higher concentration level. Will the prognosis be better, yes or no? And they compared it for non-critical ill patients and critically ill patients, and as well for patients undergoing so-called renal replacement therapy, which they do usually when they have acute kidney failure. So you need to replace the function of the kidney. So what they actually saw is that the prognosis becomes better if you have higher concentration levels. I mean, you could argue, yes, of course, if you have a higher concentration of your antibiotic in your bloodstream, yeah, of course it will be easier to eradicate the pathogen. But at that time, it was not so well known that for the most beta-lactams with the regular dosage, you won't even achieve the concentrations which are required for treatment. So what nowadays has become very popular are so-called antibiotic stewardship programs. So, for instance, in Germany, we see that almost every hospital, which is a little bit large and maybe not located in a very small town, that almost in every kind of hospital you have some kind of local antibiotic stewardship program. So those kind of programs, they simply try to get a better outcome for the patient, to reduce the resistance rate, and you typically have a team which is composed of an infectiologist, a specialist in microbiology, a pharmacist which knows about toxic effect of the effects of your drugs, and which can also aid in the administration, and also a hygiene specialist. So I and other people which are doing the TDM services, we are not actually a core member of antibiotic stewardship teams, but we try to do our best to get them the results they really need, so to get them the quantification results they need. So the WHO has also launched a global action plan on antimicrobial resistance, and here they also clearly state that the antimicrobial agents we nowadays have available, that they should really be preserved and the usage should be optimized. So this kind of global action plan of the WHO, it has been implemented in regulations on the EU. For instance, in EU guidelines, here you can see the EU guideline for prudent use of antimicrobials in human health, and this also has been implemented in national strategies and national guidelines, as you can see on this slide. So every state or every country or many countries nowadays have national strategies which focus to prevent the spreading and dissemination of antimicrobial resistance. Here again, just to show you about the antibiotic stewardship, what is the benefit? Well, we simply want a better health, a better outcome for the patients, and we simply want to prevent the emergence of resistance. And what is also essential to say is that there's data in literature that so-called antibiotic stewardship programs, that they are cost efficient. So please consider that the TDM is just a part of this entire process. Most antibiotic stewardship, it's about, well, what's the local resistance pattern in the hospital, how many drugs are being consumed, what's the outcome of the patient, and so on. So it's not just TDM. So TDM is a part of ABS. It's a minor part, but it's still an essential part of the entire process. You can see it also in the antibiotic stewardship recommendations by the Center of Disease Control, where they clearly state that therapeutic drug monitoring of antimicrobials is essential to optimize the therapy of highly drug-resistant bacteria, and they also state that it's essential to do, especially in critically ill patients, and also for the beta-lactam antibiotics. On this slide, you see an extract from the German guideline, and they also say the same, that the TDM, especially of the beta-lactams, it's reasonable and recommended in critically ill patients, especially to prevent underdosage, and they also state that programs for dose optimization are cost effective. But what we actually really lack is a clear recommendation. When should it be done? A clear statement in the guideline, who should receive it, when should it be done, and what to make out of it. So we still lack some kind of consensus guideline, as we have, for instance, for psychopharmacology, and we lack this kind of consensus guidelines for the antibiotics. So what do you need to consider when you want to set up a ABS program in your own hospital and when you want to perform TDM of antibiotics? First of all, it's really essential for you to know which are the antibiotics which are prescribed locally. For instance, you here see a table from the German SARI initiative, which is the surveillance of antibiotic use and resistance in intensive care units. So it's essentially necessary for you to know which antibiotics are prescribed, which antibiotics are used, and why is it worth to perform a TDM. Yes or no. For instance, at our hospital, I have highlighted the antibiotics we perform TDM in green, especially piperacillin and meropenem are the most common antibiotics where also the physician states it's worth to perform TDM. So when should TDM now be performed? When you have a patient and you suspect an infection, what is being done usually is that in the ABS team you have some kind of discussion. Well, what antibiotics shall we use? What kind of infection are we facing? You can also think of, let's say, a gastrointestinal infection. You have other pathogens which are the causative pathogen when you compare it to infection of the kidney. So it's also essential to select your antibiotic depending on the site of infection. What the physician is going to do, he's performing usually a so-called empiric treatment, which means he is going to perform treatment with an antibiotic he thinks is best for the causative pathogens, which are usually associated with such kind of infections. But of course, he doesn't know which is exactly the causative pathogen. Once you have all this kind of data, resistance pattern, the causative pathogen, then it's worth to perform a therapeutic drug monitoring. Why? Because you simply want to see, are my concentrations in the bloodstream, are they adequate or not? Whenever you see a toxicity, you should consider dose adjustment. Whenever you see insufficient concentration, you should change your dosage regimen or increase the dosage. So here, therapeutic drug monitoring is being performed, not before. So it's not performed initially whenever you are giving a drug, but only once you have identified the pathogen and thus its resistance pattern. As I said before, should the pathogen should you not be able to get an MSC of a pathogen or be able to identify it, then you can use the ICAS breakpoint tables. So who should receive TDM? What are the patients which should receive TDM? Well, as a rule of thumb, you could simply say all those patients with severe life-threatening infections. Yeah? So especially also those are patients at intensive care units, but also obese patients, children, patients with renal insufficiency, and those are especially patients which are at risk of achieving only subtherapeutic levels. So this is just a recommendation here by the authors of the studies, but what we as physicians really need are guidelines, national guidelines, which clearly state which patient should receive a TDM and which one should receive no TDM at all. So what we also have to consider from the practical aspect is, well, are you using a concentration-dependent antibiotic or are you using a time-dependent antibiotic? So you have to decide, do I need a trough level or do I need to measure the peak level? Or maybe do I need to measure the AUC? So whenever you are performing TDM, you have to consider those facts, and which is also very necessary. We generally recommend their physicians to perform TDM with at least 2 samples. Why is that? Because with the pharmacokinetic models we have available right now, even if you have 2 samples, you will be able to get a better pharmacokinetic curve, or as you see, for instance, on the right side, when using single samples. So we usually say them, okay, do it at least 2 times. So some practical aspects as well. This is especially important for the physician. Of course, you need the patient data. You need to know when was the blood drawn and when was the antibiotic administered. You need to know what kind of dosage regimen was it, how long was the infusion time. When we face the analytical aspect, it is really essential to keep the chain short and cold. Why is that? Because data has shown that beta-lactams are very unstable. So whenever you want to perform a TDM of beta-lactams in your own laboratory, you should prepare calibrators and QCs for your assay under cold conditions. What is also very key for analysis is the pre-analytical aspect. You need to keep your samples cold. So the best way which you could have is that the blood which is being transferred to your laboratory, that it's cooled in ice water. Please don't freeze it, because then you will get hemolyzed, but cooling it in ice water, and obtain the serum or plasma as soon as possible and store it if you can't measure it right away. So we prefer storage at -80 degrees, but our data has also indicated that storage up to 3 days at -20 degrees is possible. What is really essential for the physician is the turnaround time. So there have been many studies where they showed what was the turnaround time of TDM. So from my personal perspective, and what also literature is saying, is that whenever you have a turnaround time over 24 hours, it's inadequate. I mean, I have seen it all when talking to physicians. They simply don't care, because it's simply the time point of the blood draw, so the concentration which was measured for the kind of time point. I mean, time has simply passed, and we have a new situation, because things in critically ill patients can turn rapidly. So what we are aiming for at our institute is a turnaround time within 6 hours. The reason why we do so is that for many antibiotics, such as beta-lactams, you have a dosage 3 times a day, which means that between each dosage, when you get your result, you simply can adapt your dosage regimen, or even the amount of antibiotic you are going to administer. As I have said before, what you also need to consider when you're going to try to set up a method is sample stability. So you should obtain your sample from the physician, from the patient, as soon as possible, and also process it as soon as possible. So for instance, meropenem, it's really unstable at room temperature. It will degrade under your hands. So get your sample as fast as possible. When you can't assay it within a certain period of time, store it stable, preferably at -80 degrees, or as we have shown in our own data, you can store it up to 3 days at minus 20 degrees. But whenever possible, I highly recommend to assay the samples as soon as possible with a turnaround time less than 6 hours. What also must be considered for TDM is the protein binding. So antibiotics have a certain degree of protein binding, which means whenever you have an antibiotic bound to a protein, it simply can't exit the blood vessel system and can't reach the site of infection. So this is also a goal for the future, especially the measurement of the non-protein bound fraction of antibiotic, which is considered the biologically active fraction. So we need to, there is a need for direct quantification of those kind of unbound antibiotics. There's one method which is the gold standard microdialysis, but I mean, if you perform it in vivo, this is simply cannot done before patients. But what laboratories actually could do is the ultrafiltration process in ex vivo, which means you're trying to separate your free antibiotic from the protein bound using a molecular weight cutoff filter and centrifugation. There are certain models for protein binding, but I mean there are so many factors which influence protein binding. So I really recommend you to perform ultrafiltration whenever it's possible. I have told you previously that the target attainment for our concentrations in our laboratory are higher than 4 times the MIC. Why is that? Because there's evidence in literature when you have a higher concentration of antibiotic in your bloodstream, you will also be able to suppress resistance. So this is the main reason why I simply focus at concentrations which exceed the MIC of a pathogen by the 4 to 5-fold. So it's the recommendation by our physicians, and well, that's the way how we handle things in our own hospital. So let me get to the last point. I think I'm a little bit short on time, the quantification techniques. So when I'm talking about techniques for quantification, well, also antibiotics, we have immunoassays, or generally in laboratories, we have immunoassays with the usage of antibodies, and we have chromatographic systems, either HPLC alone or liquid chromatography, or UPLC or HPLC combined with mass spectrometry. So those are very common techniques which are used in a clinical laboratory. So what you will find in almost any kind of laboratory, clinical laboratory, is the immunoassay. I mean, it's easy to implement. The handling is very easy. You can perform measurements daily, also 24 hours each day, and you have some kind of feasible decentralization. So it's a rather widespread technology, which unfortunately is not available for the beta-lactams antibiotic. I need to state that it's available for glycopeptides and aminoglycosides, but not for the beta-lactams. Another technology which is available in other laboratories, for instance, is the HPLC. The HPLC will simply separate analytes depending on their retention on a chromatographic column, but we have the problem of rather long run times. And the big problem of the HPLC, according to my own opinion, is the detection of unspecific interfering compounds. Just imagine when you have a very ill patient, those patients from time to time, they receive over 20 or even over 30 different drugs. Those kind of drugs are all being metabolized. So you have a very complex matrix. You have a big risk that you could have interfering substances which could also interfere with the measurement of your specific antibiotic. But, which is rather good for us, recently commercialized IVD kits are available for the measurement of beta-lactam antibiotics. A method which must be considered as a method with higher metrological order is, of course, the mass spectrometry. So, just to show you, the mass spectrometry is a method which simply selects ions or molecules based on their mass to charge ratio. and you cannot combine only one mass spectrometry cell, but you can perform a so-called tandem mass spectrometry analysis, where we combine different cells of your mass spectrometer, and you are able to select analytes depending on the mass to charge ratio. You can select them within the first cell, you get rid of the other compounds. In the second cell, your analyte is being fragmented, and then you can select the daughter ions of this analyte. So tandem mass spectrometry is a method with a very high metrological order. It has a big selectivity, but it's extensive, it's also expensive, and is only available in specialized laboratories. So for instance, in Germany, we have HPLC tandem mass spectrometry in most cases only available in private laboratories or on university hospital laboratories. So let me show you right now where is TDM of beta-lactams being performed. So most countries where it's being performed are in Europe, but it's especially France, Germany, Belgium, also in Italy. It's also being performed in the States, but you could simply say that the country where you have the pioneers of beta-lactam antibiotic TDM is simply Australia. I want to emphasize the group of Professor Jason Roberts, who has done outstanding works in the field of beta-lactam antibiotics and who is the pioneer on this field. So let me get to the last point of this current presentation. Here again you see a slide of beta-lactam antibiotics of the current ring trial in Germany, what kind of methods are being used, and what you see again is that meropenem is the substance which is quantified in most laboratories, followed by piperacillin, and the method of choice is LC-MS-MS. So, what are the bottlenecks for antibiotic TDM? What are the future challenges? So, what we actually need is we need additional TDM services. As I've told you, antibiotics, they are quite precious substances. We need to preserve the efficiency as long as possible. One way to do so is, of course, the antibiotic TDM, also to prevent subtherapeutic levels, which will fuel the spreading of resistance, and subtherapeutic levels are also associated with a worse outcome. And if we have more available TDM services, this of course will fuel academic research, and this will also fuel the development of corresponding guidelines. So what we actually need, we need IVD kits. I'm telling this also with regard of the new European Union in vitro diagnostics regulation. So we need really IVD kits for the measurement of antibiotics, of beta-lactam antibiotics, but the industry should also focus on antibiotics which are now considered second line or even third line, because those are the antibiotics which have not been used that extensively and which have very low resistance rates, and these ones, one day maybe they could become common targets for TDM. What we also need are, of course, ring trials. What we need are affordable automated solutions, which should be from the level of expertise you need to handle those kind of devices, be almost similar to the immunoassays. So even if we're performing a quantification with the CMSMS, this should become much easier. We need automated version, which a regular technician assistant is also being able to handle. But of course, we need also our reference methods for antibiotic TDM, especially the beta-lactams, and what also should be considered is the measurement of the free antibiotic concentration. As I have said before, we don't have only bottlenecks with the TDM service, but we need also additional academic research. So, I mean, there are a few studies which say, okay, antibiotic TDM, it's really, it has proven itself as method to simply reduce resistance and save cost. But unfortunately, I need to say that not every kind of study is confirming this effect. So we see in certain studies that mortality and outcome are mortality is being reduced, and we observe better outcomes in certain studies, but others don't observe that. I mean, in this kind of meta-analysis you see here on this slide, this kind of meta-analysis states that there seems to be a beneficial effect, but what we actually really need are more studies, more prospective studies, and we can get only those kind of studies if we have adequate TDM services available, with uniform, also with traceability of the material which we are using for calibration and generation of quality controls. So we really need affordable, decentralized solutions so that we can generate more data. If we generate more data, I'm sure we will be able to generate more data, to obtain better recommendations for the physician for his daily struggle with increasing antimicrobial resistance. So my concluding remarks for this talk of today are the following ones. So for the community, it's a big effort to implement the resources for antibiotic TDM, and I think really that's a big challenge for laboratory medicine, but also for public health and industry. But I think it's worth, because there's a wealth of data that suggests that when you're performing TDM, when you're performing it properly, that you get a significant improvement for the outcome of patients, that you can increase their prognosis, and that it also have cost-effective programs which will be can be implemented when you perform antibiotic TDM. And I really personally think that mass spectrometric methods, that they are key, and that they will play a major role in this context, because those are the methods of the highest metrological order. We need a high selectivity whenever we perform antibiotic TDM, because the patients where we're performing it, those are simply the ones which have the most complex matrices. And here I really think that mass spectrometry is the key technology to do TDM. So I'm
right. That's very interesting. Thank you very much, Dr. Pahl, for your presentation. I personally found the presentation very thought-provoking and useful, and I hope that you, the audience, also found it of value. I know that there's hundreds of people on the call, and there's lots of questions coming in. We're not going to have time for all of them, but we'll have time for many of them. So the first question I'd like to ask you is, which patients are at risk of sub-therapeutic beta-lactam concentrations and therefore require closer monitoring for your TDM?
Could we jump to a specific slide?
If you want to, I don't know. If you want to, sure, go to a slide, if it's helpful.
Yeah, of course it is. I mean, it was the slide number 29. Well, actually, you could simply say that it are the patients which are critically ill. So, for instance, we at our wards perform it for patients which have multiple organ failure, which have kidney failure, which have liver failure, because when you think of the antibiotics, those are also often cleared by the kidney, or others are also cleared by the liver. So generally could say critically ill patients, but there's also data that you should perform it in the elderly, because they have a changed volume of distribution, but also there's data that you should perform it in obese patients or also in children. And whenever you also are using an antibiotic where you say you have a rather tiny penetration into the tissue, that's also clearly recommended to perform, because you need rather high levels to obtain a concentration to fight the causative pathogen at the local site of infection. So I highly can recommend the work by Huttner et al. And Müller, which clearly states which are the target populations for antibiotic TDM.
Okay, and another question is why is it difficult to establish uniform target ranges for beta-lactam antibiotics?
Yeah, the problem is we simply have a lack of prospective data. I mean, those kind of targets I mentioned before, they are almost all of them are mainly derived from in vitro studies and animal models. There have been a few studies which confirmed those kind of targets, but to be honest, nobody is sure what is simply the best target. So we at our ward, we better play safe than sorry, and that's also another reason why we are aiming at fourfold MIC concentrations. But actually nobody knows, and for that we really need good prospective
Exactly.
studies.
Lots of studies going on to establish some sort of a guidelines worldwide, of course. Yes.
Exactly.
Yes. What should be the lowest limit for the target range in cases where there's minimal inhibitory concentration is not known? Do you use the clinical breakpoints or something entirely different?
Exactly. We in this case use the clinical breakpoints from the UCOST document. Exactly. That's the lowest concentration level you should actually achieve. Yep. Exactly.
And is it worthwhile to measure the free antibiotic concentration? And if so, what's the best method of choice for clinical labs to use?
I personally think it's the ultrafiltration. I mean, so you can get those kind of molecular weight cutoff filters, but what you have actually to consider whenever you are performing ultrafiltration, you need to perform a good validation process, because, I mean, you have to exclude unspecific binding of your antibiotic to the filter. But I really think ultrafiltration is the way to go. And I also know other people which are measuring the free concentration. They all do it with ultrafiltration.
Okay, fine. And so you were very enthusiastic about mass spec. So what is the potential of mass spec analysis for antibiotic TDM in the future?
Well, for me the potential is clearly that the selectivity of the assay. I mean, as I said before, the critically ill patients, they receive a lot of medications, and whenever you think that a new medication is going to be approved, of course, it is going to be administered to a patient, of course you have to think of selectivity, right? Because if this kind of new drug would interfere with your assay, yeah, well, then you would measure maybe false high values of your drug. So since mass spectrometry is of the highest metrological level, and as you have a rather high selectivity or a very high selectivity, I simply think that's the only way to go. Yeah.
Yeah, fine. Someone else was saying to lower the threshold for clinical lab managers to start using mass spec, are there any central public data banks of resistant bacterial results to help them get started?
So when a person wants to start to perform antibiotic TDM, is this your question? If there's any kind of...
That's right. But is there any sort of, rather than just going to do the whole thing themselves, can they actually plug into information that's coming from, say, some central public set of results to help them get up to speed quicker?
Unfortunately, I by myself also haven't found any kind of central database where everything is being stored. So whenever somebody wants to establish a new assay or wants to implement it, there's the local guidelines you could use, but also there's simply the literature on the internet, which you have to read and go through.
Right.
Yeah.
So perhaps there's a need to have this set up somewhere by someone
to
help everyone out there. Yes.
Yeah, of course.
Yes.
And you also need to generate and provide this kind of structure also in, let's say, in a national setting, when a hospital says, okay, guys, I want to implement a new antibiotic stewardship program, and I really need TDM in my hospital because I have problems with resistance. There are some places from the government, but from my point of view, it's really insufficient so far. So there's a lot of effort which is still necessary for the physician or for the people at the local hospitals.
Yes. Okay. So someone else was asking about, we know that AKI can happen in critically ill patients because of different reasons, although patients may have no history of renal complication. How can we adjust antibiotic dosage in critically ill patients with AKI, especially if they have sepsis, or is that a bit too specialist a question for you?
What you can do actually is, as I know also from the physicians from our intensive care units, you can, for instance, adjust it, or you can reach sufficient levels when you change the way of application. So there has been a lot of data where they, for instance, compare bolus regimens where they perform a rather fast infusion of an antibiotic compared to a so-called prolonged infusion, where you're decreasing the flow rate of your antibiotic entering the body. But there's also data being generated about so-called prolonged continuous infusion regimens. So I think switching from the traditional bolus regimens, where you say, okay, I administer one dosage every 8 hours, maybe changing to so-called continuous infusion settings where you are administering antibiotic continuously. I think this is a very interesting approach and one way to go, because when you infuse it continuously, it's clearly from the pharmacokinetics you are probably will be able to reach the trough levels in a more efficient way when compared to bolus regimen. So I think this is a very interesting approach, and there's also some literature on that, changing the dosage regimen, so the way of administering an antibiotic.
Okay, interesting. A related question. Someone was saying, you're talking about continuous dosing. Are there any biosensors being worked on for a good solution for TDM? Because that also might be a way, also in terms of helping for patient control and so on.
Mm-hmm. Well, actually, some biosensor. I'm not aware of any kind of biosensors for the TDM itself, but what I know is that according to the guidelines we have right now, we use the biomarkers for infection control, which means, let's say you have a acute phase protein, or you have a biomarker like procalcitonin. Those are really valuable markers which will simply indicate that the pathogen load in your body and the inflammation is simply decreasing. But I by myself, I'm not aware of any kind of other markers which could be used as something which could replace the antibiotic TDM. But there are certain biomarkers which are used for treatment to check whether your infection is where you are obtaining a cure with an antibiotic, yes or not.
Yeah, interesting. Time's running on. Time for a couple more questions. Someone was asking when there is a PK/PD optimal value for antibiotics such as imipenem or the like, are there any maximal concentrations to avoid neurotoxicity?
Yes, that's true. There are certain concentrations which should be avoided, but this is not. But that's not such a problem for meropenem. Well, especially not in our hospital, because when I see the concentrations, I never have seen any kind of very high concentration. But yeah, for the beta-lactams, you also have some kind of concentration levels which are considered toxic. But from my point of view, I would say that this is most the issue for cefepime. But meropenem, from my own experience, it's not that troublesome. But of course, whenever you're measuring, you need to also consider toxic concentration levels.
Right. Okay. And I think we've got time for one last question. It's so how can we promote antibiotic TDM in developing countries who have limited resources? Is there any particular route, any particular way of thinking about this?
Yeah, that's a very interesting question. I mean, I can imagine that it's rather difficult to promote LC, MS solutions for developing countries. One way to do so, I really think, is when we talk of the analytical aspect, then I would go for the HPLC system. HPLC systems are rather inexpensive. They are, when you have an available commercialized kit, that's of course the way to go. But it's not just, as I told before, it's not just about the TDM. When we want to fight the spreading of antimicrobial resistance, but a very good and very effective aspect is the antibiotic stewardship. So I would say that for countries with limited resources, this is would be a very efficient and cost-saving process if they established antibiotic stewardship programs where they simply try to improve the patient outcome by monitoring administration of drugs, checking local resistance pattern, and I think this is the way to go. When it comes to the analytical aspect, I think HPLC would be a affordable solution.
Okay, fine, thank you. We obviously don't have time to answer all the questions that have been sent in. So thank you, the audience, for your participation, submitting some great questions. Any questions we weren't answer today due to time, then our speaker Michael will try and contact you later with an answer directly, because you all have your email addresses. I'd like to take this opportunity to thank again Dr. Michael Powell for his presentation, also you, the attendees of today's webinar, and I trust that you, as I, found this a very informative webinar with lots of thoughtful questions and answers. And please remember that there's been a recording of this webinar made available online shortly, all the slides, all the questions, all the answers. So thank you, everyone, and I hope you have a good day. Goodbye.